The role of genetic engineering in enhancing food safety. Title: The role of genetic engineering in enhancing food safety. Introduction. In recent years, genetic engineering has emerged as a promising technology with the potential to revolutionize the field of agriculture. One of the key areas where genetic engineering holds tremendous promise is enhancing food safety. Through the manipulation of an organism's genetic material, scientists can develop crops that are more resistant to diseases, pests, and environmental stressors. His article explores how genetic engineering plays a crucial role in ensuring a safer and more secure food supply for the growing population. 1. Genetic engineering and crop disease resistance. Crop diseases pose a significant threat to global food production. Traditional breeding methods have often fallen short in producing disease-resistant varieties, leaving crops vulnerable to pathogens. Genetic engineering allows scientists to introduce specific genes into crop plants that enable them to resist or tolerate diseases. For instance, the use of Bt genes in crops like corn and cotton has greatly reduced the need for chemical pesticides by providing built-in protection against insects. In this way, genetic engineering helps ensure that our food is safe and free from harmful chemicals. 2. Pest control and genetic engineering. Pest control is another critical component of ensuring food safety. Invasive pests can cause devastating damage to crops, leading to economic losses and food scarcity. Genetic engineering offers a powerful tool in developing crops that are naturally resistant to pests. The introduction of genes from bacteria or other plants into crops provides built-in defense mechanisms, eliminating or reducing the need for pesticide applications. By decreasing pesticide usage, genetic engineering contributes to a safer and healthier food supply, reducing environmental contamination and potential human health risks. By introducing specific genes, scientists can enhance nutrient content, making crops more nutritious and thereby promoting global food security. For instance, golden rice, a genetically modified crop enriched with vitamin A, has the potential to address vitamin A deficiency, which affects millions of people worldwide. By genetically engineering crops with higher nutrient levels, we can reduce malnutrition and improve overall public health. Genetic engineering offers a pathway to develop crops with increased tolerance to these conditions. Scientists can engineer crops to withstand drought, salinity, or other adverse conditions, ensuring higher yields and better food safety in regions prone to these stressors. By harnessing genetic engineering's potential, we can secure food production systems against the uncertainties of a changing climate. Governments and regulatory bodies play a pivotal role in establishing robust regulations and guidelines for the safe development and use of genetically modified organisms, GMOs. Rigorous testing and evaluation are necessary to assess the potential risks to human health and the environment. Moreover, public engagement and transparent communication about genetic engineering technologies are vital for building trust and acceptance within communities. One major ethical concern is the potential for unintended consequences. Genetic engineering involves manipulating an organism's genes, and there is a risk of unintended effects on the environment and ecosystems. Additionally, there are concerns about the long-term impact of genetically modified organisms on biodiversity. Another ethical consideration is the potential for monopolization. Furthermore, there is a debate surrounding the labeling of genetically modified foods. Striking a balance between transparency and consumer choice is crucial in navigating this ethical issue. Livestock diseases can have detrimental effects on animal health, welfare, and food production. By using genetic engineering techniques, scientists can introduce genes that enhance the animal's immune systems or confer resistance to specific diseases. This approach not only reduces the risk of disease transmission but also minimizes the need for antibiotics or other treatments that may leave risks. I-DOs in the food chain. By incorporating genetic engineering into livestock breeding programs, we can create healthier and safer food products. 8. Ensuring allergen-free foods. Food allergies are a growing concern worldwide, with potentially severe consequences for affected individuals. Genetic engineering can play a role in producing allergen-free foods by removing or reducing the allergenic proteins in certain crops. For example, scientists have developed genetically modified soybeans with reduced levels of allergens, providing a safer alternative for individuals all. Allergic to soy. By utilizing genetic engineering techniques, we can mitigate the risks associated with food allergies and provide safer food options for consumers. 9. Controlling mycotoxin contamination. Mycotoxins are toxic substances produced by certain fungi that contaminate crops, posing serious health risks. Traditional methods of mycotoxin control, such as fungicides or physical removal, may have limitations or undesirable effects. Genetic engineering offers a potential solution by developing crops with improved resistance to mycotoxin-producing fungi. Scientists can introduce genes that enhance the plant's defense mechanisms and reduce mycotoxin levels. By reducing mycotoxin contamination in crops, genetic engineering contributes to safer food production and consumption. 10. Addressing antibiotic resistance. Antibiotic resistance is a global health concern that can be exacerbated by the use of antibiotics in livestock production systems. Genetic engineering provides an opportunity to address this issue. Why developing livestock that are more resistant to diseases without relying on antibiotics? By introducing genes that enhance immune response or confer resistance to specific pathogens, scientists can decrease the need for antibiotic treatments in animal agriculture. This approach helps safeguard the effectiveness of antibiotics in human medicine and promotes food safety by reducing the risk of antibiotic-resistant bacteria in the food chain. 11. Increasing food shelf life. Another area where genetic engineering can enhance food safety is by extending the shelf life of perishable foods. Many fruits and vegetables are prone to spoilage, resulting in food waste and potent health risks. Through genetic engineering, scientists can modify the genes responsible for the deterioration of food, such as enzymes that cause ripening or decay. 12. Developing all-weather crops. Genetic engineering also offers the potential to develop crops that can thrive in various climatic conditions. Extreme weather events, such as droughts or floods, can have devastating effects on crop yields and food availability. By introducing genes that confer tolerance to such conditions, scientists can create all-weather crops that are resilient and less vulnerable to climate-related challenges. These genetically modified crops can ensure a stable food supply even in regions with unpredictable or harsh climates, contributing to food safety and security on a global scale. 13. Enhancing food traceability. Genetic engineering techniques, such as DNA barcoding, can facilitate improved food traceability and identification of potential safety concerns. By implementing unique genetic markers in crops and livestock, it becomes easier to track and monitor the origin of food products throughout the supply chain. This increased traceability allows for quicker and more effective recalls in the event of foodborne illness outbreaks or other safety issues. Genetic engineering, therefore, plays a crucial role in ensuring that food safety regulations are adhered to and that consumers have access to safe and traceable food products. 14. Advancing detection and diagnosis of foodborne pathogens. Genetic engineering has also contributed to advancements in detecting and diagnosing foodborne pathogens. Traditional methods of identifying bacteria or viruses in food samples can be time-consuming and sometimes inaccurate. This allows for early intervention and effective control measures to prevent outbreaks of foodborne illnesses, thus enhancing food safety and protecting public health.